Going to get started and continue on our journey of understanding microbial pathology and virulence and different virulence factors and how to identify them and test them. And I forgot my major point for today, so I'm sorry about that. So I'll just point at the screen. So we're talking about host pathogen interactions and how bacteria actually cause disease. At the molecular level, that's what we're going to be talking about in future lectures. But today is sort of more of a zoomed out view of infection and infectious processes and resulting disease. If disease does result from an infection, you can quantify that. Okay, and so this is something you might be interested in doing if you are a pathologist and you're trying to understand levels of virulence or infectivity and resulting disease of a particular group of microorganisms. Okay, so there are two things you can do to help quantify this. You can quantify what we call the ID50, which is the infectious dose, or the LD50, which is the lethal dose. Okay, so I'll talk about the differences between the two of those. And the infectious dose, or the ID50, is the number of organisms it takes to colonize 50% of your hosts. Okay, so you would infect a group of hosts. So these would all be the same hosts. So for example, if you're working in corn and you're trying to understand the infectious dose of a particular strain of bacteria that affects corn, you would say have 100 corn plants. And then you would be testing different doses of the bacteria in sets of 100 corn plants. So imagine this is a lot of plants. So you would have, for example, 10 bacteria that you infect with this first set of 100 plants, 100 as your dose for the next 100 plants, 1,000 for the next 100 plants, and so on and so forth, generally by degrees of multiples of 10. Okay, so then you would look at those groups of 100 plants and you would look to see where 50% of those plants got infected. Okay, so for example, 10 to the third, your 10 to the third dose was your one where you saw 50% of the plants got infected. That would be your ID50, or your infectious dose. So it was the dose of bacteria that it took to colonize 50% of your test subjects. Okay, so that would be 50 of those plants were infected with the bacteria. It doesn't mean that they actually have to, so it means the ones that also infected but also caused disease symptoms. Okay, so that would be a visual cue. So you would see 50% of them got sick with your dose of bacteria. So that's your ID50. So that's just showing symptoms, right? So that's not killing the plant. That's just showing symptoms. So if you wanted to figure out what the lethal dose was, you would do an LD50. So you would do the same experiment with the same setup of different doses of your bacterial inoculum, and then you would look to see not just where 50% of the plants got sick, but where 50% of the plants got killed. Okay? So your LD50 is usually higher than your ID50. So it takes more bacteria to actually kill a plant than it does to infect and cause symptoms. Okay, so we said our example would be 10 to the third for our infectious dose. It might be 10 to the fifth as the lethal dose. Okay, so from this graph you can look at here, you can look at the doses of organisms administered per test subject. In this case, they're talking about animals and not plants. You can see that they're testing agent 1, or microbe number 1, and agent 2, or also known as microbe number 2. And you can see that the LD50, or the lethal dose for the first agent, for which they kill 50% of the plants, is 400 units, or 400 cells. Okay, whereas the LD50 for the second agent is 600. So agent 1 is more virulent than agent 2 because it takes fewer cells in terms of a dose of inoculum to kill 50% of the population that you're testing. Okay, so that makes sense, right? The fewer number of cells that it takes to inoculate, as compared to agent 2, means that you have you're more virulent if you have a lower LD50. And remember, the difference between infectious, or ID50, or lethal, and LD50 is infectious dose versus lethal dose. So infectious dose is just the amount that it takes to cause symptoms, and lethal dose is the amount that it takes to kill 50% of your hosts. Okay, so just going over what I just said in the graph. So a low LD50 means that few microbes are required to kill a specific host, okay? So that means it's highly virulent. If you only need to give it, say, 100 cell units and it can kill a host, that's pretty crazy, right? That's a really low LD50. And if it's a high LD50, that means more microbes are required to kill that host that you're inoculating, okay? So a microbe with a low LD50, like we talked about agent number 1 in the graph before, is more virulent than a microbe with a higher LD50. And in general, the LD50 is higher than the ID50. Does that make sense? So you're testing lots of different doses of inoculum in sets of plants or animals that you're testing. And from that, you can derive your ID50 or your LD50. Okay? So now we're going to move on to infection cycles. So that was something that we did on purpose in the laboratory, right, to infect our plants to get our ID50 and our LD50. But the infection cycle is what happens out in nature. Okay? So how do microbes move around from one organism to another and colonize that particular organism? Like, how the heck did I get this virus that I've been battling for the past week? Who knows? But there are different ways that could have happened. And so basically an infection cycle is just the ability of a pathogen to spread within a population from host to host. Okay? So that could be by different ways. It could be direct contact, like someone could have sneezed on me, like my child did. Or I could have gotten indirectly infected by touching, say, a grocery cart that someone had sneezed on, and then I touch my face and I infect myself that way. So there's direct and indirect modes of infection in the infection cycle. Okay, so the indirect contact, well, direct contact is very self-explanatory, right? You have to directly contact someone who is infected with something, and that gives you that particular microorganism, and then you can develop disease depending on if you're susceptible or not. Indirect is a little bit more of a gray area there. Okay, so this could involve contact with what we call fomites. So fomites are inanimate objects, so like the grocery cart handle that I was talking about, that can be contaminated with something. A doorknob, for example, could be contaminated, and then you later go on to touch your face or put your fingers in your mouth, and that is how you acquire that particular virus or bacteria or fungus. It could be like the shower in the gym where you're going to get athlete's foot, right? So the floor of the shower, someone has deposited their tinea pedis onto that shower floor, and then you come in and take a shower without your flip-flops, and you get that particular athlete's foot infection as well. So that's what a fomite is. It's indirect contact with an inanimate object that passes that pathogen to you. Okay? We also have something called horizontal transmission, which requires a vector. So a vector is just something that carries something to another place, right? So it could be an insect. Those are very common vectors of disease. So they acquire the pathogen, for example, a mosquito. It could be harboring dengue virus or yellow fever or other pathogens that are mosquito-borne, and then it bites you and infects you. And then another mosquito could come and acquire that infection from you and then pass it to another person. Okay, so that is what a vector is. So a vector is just something that carries something from one place to another. We talked about it in terms of recombinant DNA technology, where we're using plasmid vectors to carry DNA from one place to another. This is the same thing. It's just a different way of defining vector. So it's something that's carrying something to another place. Okay? Now, you can have another layer of this horizontal transmission where you would have a reservoir so all of this is in the context of human pathology, right? So this is how we define things, is based on our own species. So a reservoir, for example, could be an animal that is carrying this particular pathogen. It may or may not be susceptible to that pathogen, but it is harboring it. And then through a series of different inoculation mechanisms, one could be insects, for example, because that can pass on to humans. Then that animal, like this bird in this graphic, is the reservoir for that. Okay? So it could be any animal, bird, or insect that harbors the pathogen, where that pathogen can propagate, and it's at a relatively high population level and serves to act as a place where this organism can propagate and then be passed to humans, right? So a very good example of that is the bubonic plague. So Yersinia pestis is the bacterium that causes the bubonic plague. It can run rampant in rodent populations, okay? And then fleas will feed on those rodents and then also bite humans, and that is how we contract the bubonic plague, okay? So the rat in that case is the reservoir, the fleas are the vector, and the humans are the unfortunate recipients that result in the bubonic plague. Okay, so how do pathogens get into you? So we talked a little bit about this last time. So there's lots of different ways of entering a human or animal body, okay? And generally they use portals of entry that are best suited to the environment that they want to colonize, right? So if you are a cold virus, like the rhinovirus, you are going to enter in through the respiratory tract, because that is where you thrive and where you do best. If you were to enter in some other way into the human body, you might not be able to propagate yourself. So that makes sense, that aerosolization, so when someone sneezes out that virus, and then it can quickly be sucked into someone else's respiratory tract, that's a very suitable way for that organism to move around within the population, okay? Foodborne pathogens, they're generally ingested when you're eating food, and so they enter through the mouth and can colonize either your stomach or your intestine and cause diarrheal diseases or stomach virus, depending on what you're talking about. I already talked about airborne. So mucosal surfaces, that is anything that can bind to the mucous membranes that are all throughout the interior of your body. That could be in your intestines, that could be in your nasal passageway, that could be in your mouth or other places. Wounds, so if something is a good wound colonizer, it's probably also very good at colonizing your skin, okay? And then you can also have this parenteral route where it's injected straight into the bloodstream. So these would be your bloodborne pathogens, and so that's why avoiding needle sticks and other things that could be contaminated with potential pathogens is a good idea. You don't want to put them right into the place where they would thrive. Okay, so we're going to talk about virulence factors. And first of all, what are virulence factors? So I've had some students ask me that. I'm going to throw that term around loosely because I use it a lot in my own vernacular, but I realize that I need to explain it better. So a virulence factor is an individual characteristic encoded by genes of a pathogen that will allow that pathogen to invade the host and cause disease. Okay? So you can have more than one virulence factor if you are an infectious bacterium. Okay? And so it's more of an additive effect. So say I have 5 different virulence factors that could be toxins or any slew of things that we're going to be talking about as virulence factors. And if I take one of them away, then I'm not completely avirulent. I just am a little bit less virulent. Okay? So you can have 5 of these, and you take 2 away, then you can have a measurable difference in your level of virulence. You could measure that by quantifying your ID50 or your LD50. Okay, that's a very good way of doing that. And so then you can start to tease apart what makes these pathogens tick in terms of their virulence factors. And so it's very exciting to us in my lab if we identify a virulence factor or a set of virulence factors that a particular microorganism uses to infect its host. Okay? And if you do identify that, you might be able to have those as drug targets or some sort of control mechanism to dampen the effects of those virulence factors. So that's why there's tons of researchers working on virulence factor identification in plants, animals, and humans. Okay? So we can do something called molecular Koch's postulates. So we know what Koch's postulates is. Okay? So that's where you are identifying the causal agent of an organism by first isolating that microbe and then putting it back into a healthy individual, recapitulating the symptoms that you saw in the first sick one, and then reculturing it out of that newly sick individual and showing that it was the same thing that you put in and the same thing that you pulled out from the first infected individual. You can do the same thing on the molecular level, right? So this is called molecular Koch's postulates. This is where you are looking at a microbe and you say, okay, I see this genome and I see these things that might be virulence factors. I'm going to remove one of those by mutation. Okay, you can do your transposon mutagenesis. And then I'm going to put it into the host and compare it to the wild type and see if, in fact, it is knocked down in virulence. And I say, oh, yes, it is. That's wonderful. Okay, then to complete molecular Koch's postulates, you have to put the wild type copy of that gene back into your mutant. Okay, and that's called complementation. So that would be the last step of molecular Koch's postulates, where in this case, in the second step, you have shown that you've knocked down virulence in the mutant, and then when you add back the wild type gene, you bring the virulence back up to wild type. Okay, so that just completes the molecular Koch's postulates for identifying virulence factors. Okay, so all virulence factors enhance the disease-producing ability of that pathogen. That is their job. Okay, and so it's all about natural selection and competition in environments. And so if the microbe's job is to acquire nutrients from a host, there are toxins that will lyse host cells and release those nutrients. And so these are things that are considered to be virulence factors. It is basically just the bacteria or the fungus or the virus trying to survive in a highly competitive environment. Okay? So they don't think of these as virulence factors, but certainly the host does, because it's making that host sick. Okay? They can facilitate attachment, so we're going to talk about adhesins that are involved in that, and so that kind of goes back to our biofilm formation lectures, where adhesion is the first step of that, and that's a requirement. They can promote bacterial growth, so the example I just gave of toxins that would lyse host cells to release nutrients to allow the bacteria or the fungus to acquire those. They can promote disease symptoms, so that lysis, for example, could be something that would be like an ulcerative situation where you're lysing the host cells and you can actually see that as part of the pathology because you have all of this dead tissue around. And some of them are even more stealth in the fact that they can help in evading the host defense mechanisms. Okay? So they can dampen host immunity and sort of circumvent that. And so then when they, while that is happening, that dampening of the host immune response, the bacteria have a chance to proliferate. So by the time that the host is caught up with everything, and say, oh, okay, actually I'm going to come back with another attack, the bacteria have already proliferated to such a population that they're going to survive within that host. Okay? Okay, so like I mentioned, that virulence factors help distinguish from an avirulent form of a species of an organism versus a virulent form. So the virulent form would possess virulence factors. The avirulent one might be lacking those. Okay? So sometimes it's very hard to taxonomically differentiate something like Yersinia pestis that has a lot of virulence factors versus one that doesn't, or E. coli that has a lot of toxin virulence factors versus one that doesn't. There are lots and lots of E. coli strains in the environment. Many of them are innocuous, many of them are already in you, but some of them have plasmids, for example, or areas in their genome that they've acquired from gene exchange that encode virulence factors. And so those are the ones that get the most press and the most research focused on them because they can cause violent diseases, right? And so it's these virulence factors that everyone is focusing on and trying to understand how that process works. Virulence factors are encoded by virulence genes, okay? So that's a very easy concept to grasp. We're going to be talking about toxins as virulence factors. We're going to be talking about adhesins that are proteinaceous adhesins that allow bacteria to stick to host surfaces. We've already talked a little bit about exopolysaccharide capsules, and we're going to talk about other things that are involved in avoiding innate immune responses from particular hosts, and also helping to evade adaptive immune systems as well. Okay? I wanted to put this back in here, this slide. You've seen this before. So we talked about the GC content of particular chromosome of a bacteria, and that's just literally counting up all of the G's and C's that are in the chromosome or in the DNA of an organism. and you get this percentage, this GC content. So 50% in this example of the chromosome of that organism is comprised of G's and C's, whereas the other 50% are A's and T's. Okay, it's a very simple concept. It's very species specific, okay? So you can see that, species 1 has a 50% GC content, whereas another one might have 42%, and that was an old school way of differentiating different species from one another when you were trying to name a new species or determine, am I dealing with the same thing or is this something new, okay? So we know that there is rampant gene exchange among bacteria, and that is not species-specific sometimes. And so they can acquire genes from other organisms that are either closely related or maybe not even closely related to them. And that can show up in the genome. And you can calculate that by simply calculating the GC content as you're going down that chromosome. And for example, this one is normally 50% GC content, and then suddenly you'll see a dip in one little area of the genome down to about 40%. And that signals that that segment of the chromosome came from a different organism when you have this little dip. And those are called genomic islands. If we're talking about virulence, these are called pathogenicity islands. Okay, so a lot of times these things are maintained within a population of a bacterial species, because they encode things that help that bacteria survive. So it's all about natural selection, okay? So this organism that has this pathogenicity island is better fit than one that might not, okay? So that's why it's maintained in the population and why we can observe it in nature, all right? So a lot of times these pathogenicity islands or genomic islands, when you look at them, they encode blocks of DNA that encode toxins or attachment things. A lot of times the type 3 secretion system will be in these pathogenicity islands. We're going to talk about what the type 3 secretion system is next time, but it's a very important secretory system that bacteria use to dampen host immune responses. A lot of times they're on these pathogenicity islands, okay? And they're transferred through, excuse me, horizontal gene transfer or HGT. Okay, so now we're going to focus on attachment, which is something that I really like to work on in my lab. We're focused on studying adhesins and what mediates adhesion, and reversible versus irreversible adhesion, and how do bacteria stick to plant and insect surfaces, and then how do they detach during some of the infection processes, okay? So some of these adhesins, there's a whole slew of them, but we're just going to pick a few popular ones. We're going to talk about pili, and pili and fimbriae are sort of interchangeable terms, and these are just extrusions from the cell, kind of like flagella, but not flagella. They're sort of hairy-like things that stick out from the surface of the bacterium, and they mediate adhesion. We're going to talk about two types of pili. We're going to talk about type 1 and type 4. But basically, they are hollow fibrils with tips that help the bacteria stick to surfaces, okay? And why does bacteria need to stick to surfaces? So there are, in the human body, for example, lots of ways of expelling microbes, okay? So this is one way that we're not always sick all the time. So we have lots of ways of doing this. So like our mucosa and our dead skin are constantly being expelled from our body. We shed a lot of skin on a daily basis. And we also shed a lot of mucosal layers through different processes that we don't need to go into from our bodies, okay? And with that, bacteria are also being shed, and fungi and things, okay? When you go to the bathroom, you're expelling liquid at a pretty high shear force through your bladder and your urethra, so that prevents infection of the urinary tract by simple physics, okay? Coughing is one way to get things out of you, and that is helping the cilia in your lungs to help propel out microbes that might be trying to colonize that area. And then when you also defecate, you're expelling a lot of your intestinal contents and a lot of microbiota during that process. So if you have a diarrheal disease, you are expelling things at a much more rapid rate than normal. And so that's one way of trying to get things out of your body, okay? So bacteria have to sort of answer back to that. And they have to say, well, all right, well, I still want to be here, so I'm going to figure out how to stick here. And so they do that by either pili or other non-Fimbriae adhesins that are surface proteins that help bacteria bind to host cells. Okay, so anything, an adhesin is just a general term for anything that promotes attachment to a host surface. Okay, so we're going to talk about these pili, and like I told you, they're sort of hair-like appendages. You can see them with a very good microscope or an electron microscope. You can visualize these things. And then we also are going to talk about a little bit about afimbriate adhesins. So afimbriate adhesins are just anything that are not Fimbriae that are involved in adhesion. Okay. All right, so we're going to do the type 1 and the type 4. I really think pili are super cool, so we're going to watch a couple of YouTube videos about pili. So type 1, they're very simple to understand. Again, they're those hair-like appendages that are produced, and then they're static. Okay, they're not involved in retraction or extrusion from the cell. They're just made, and then they're involved in attachment. Okay, so they're just like things that help bacteria stick to a surface, and that is basically their primary job. Okay, type 4 pili are a little bit more dynamic. They're also hairy extrusions from the bacterial cell, and they are produced in a similar way, but they can move, right? So they can assemble themselves and they can disassemble themselves. So they can be present or absent, or build themselves and pull themselves back in, depending on the situation. And these are involved in motility, okay? Because of that extraction, retraction, they can help bacteria or mediate movement across a surface. So these are involved in motility, and I want to stress that is different than swimming motility, which is mediated by flagella. So we talked about flagella and how they propel themselves through liquid environments, okay? So that's not this. This is a different form of motility. It's called either swarming or gliding motility, and it's surface-based. So this is how bacteria crawl along the surface. They do need a little bit of moisture, but it's nothing like an aqueous environment, like a swimming pool or something like that, where flagella would be involved in swimming, okay? So it's called twitching motility, also swarming motility, and also gliding motility, depending on which literature you're looking at. Okay, so type 1 pili, I just want to emphasize here that the pilus itself is made of repeats of the pil-A protein, okay? And so it's built, and then it's extruded out from the surface, and you can see that there's a cap protein, and there are tip proteins, but in the middle, there are pil-A subunits that are just repeats of the same protein, over and over and over. Type 4 pili are similar, and also want to mention that once this is built, it's static. It doesn't retract and extend like the type 4 pili. Again, pil-A, a protein that's called pil-A, which is different than the pil-A in structure from the type 1 pili, also make up the primary component, or the pilus, of these type 4 pili, okay? So this can extend and then retract based on the amount of pil-A subunits that are either being deposited into the pilus or subtracted from the pilus, okay? And this can happen very, very quickly. In matters of seconds, you can build and retract, okay? So we're going to watch an animation that shows us that. But first, I just wanted to show you a time lapse of what goes on during type 4 pili-mediated motility along the surface. So it's basically analogous to what the textbook refers to as Spider-Man motility. So the bacteria sends out this pilus, okay, so they're building it by adding pil-A subunits. It's sticky on the end, so that sticks to the surface. And then as it retracts, it's pulling itself towards that stuck tip of the pilus, and then it's disassembling during the retraction. So then it will send it out again, and it will stick to a surface, and then through that retraction, it will pull itself towards. So it's like Spider-Man motility when he sends out his web and he pulls himself towards a building with his web that shoots out his arm. Okay, so the web is like the pilus, and then the retraction is like the pulling. So it actually looks like twitching because it's kind of, it's not very synchronous, and it's the bacteria just pulling itself through all its pili across the surface. Okay, so I'm going to watch an animation that shows you that. And there are a lot of bacteria that can do this. One of them is Xylella fastidiosa, which is what I work on, and it's actually featured in the animation, so I was very proud to see that. Pseudomonas aeruginosa is also a very good twitcher, and so a lot of different movies have been made showing twitching motility and all these different bugs. Okay, so
a broad spectrum of gram-negative bacteria has the ability to migrate across a moist surface without the aid of flagella. The bacteria on this agar surface are moving by a mechanism called twitching motility. Twitching motility requires the presence of a type of filament called a type 4 pilus. Type 4 pili extend from one or both poles of a bacterium. The cells use a type 4 pilus like a grappling hook, extending a pilus, sticking it to a surface, and then retracting the pilus, which pulls the cell forward. Here's a repeat of the sequence. Type 4 pili allow bacteria to stick to inanimate surfaces. The pili also mediate the attachment of bacteria to each other during the formation of biofilms. The pili also mediate the attachment to host cells, and for this reason are important in pathogenesis. All type 4 pili use similar secretion and assembly machinery involving at least a dozen proteins. The pilus fiber itself is composed of many subunits of a protein called PIL-A. PIL-A originates in the inner membrane as a slightly longer protein called pre-PIL-A, which gets clipped by a protease called PIL-D to form a mature PIL-A protein. During elongation, the PIL-A proteins are transported from the inner membrane through a channel called PIL-Q in the outer membrane. The assembly of the pilus requires the hydrolysis of ATP at the inner membrane by a protein called PIL-F. As PIL-A subunits add to the base of the pilus elongates. The retraction of a pilus also requires the hydrolysis of ATP, this time by a protein called PIL-T. PIL-A subunits are removed from the base of the pilus, and with each subunit removed, the pilus shortens a tiny bit. It has been estimated that the pilus retracts by 1,000 to 1,500 subunits per second.
So the animation is very slow in showing you that, but just to remind you that this can happen very, very quickly, as you saw in the animation. Okay, so I pulled up some other videos because I have a lot of friends or colleagues that work on twitching motility, so I wanted to showcase some of what I learned at conferences and also what my colleagues are doing. So this is Pseudomonas twitching, and you'll see here is sort of like the zoomed out view, and then there is the close-up of what's in the square. And you can see that's a very coordinated effort. So there's a lot of communication that goes on, and like quorum sensing type things that help mediate this, and it kind of looks like traffic on the LA freeway, right? So it's very organized, but also chaotic. So you can see that they're moving and they're following certain patterns, and there's some that are going this way, some that are going that way. And this is on an agar surface. This is a solid surface. Okay, so this is not swimming motility. This is twitching motility. Okay, so that's that. And then I found another one to sort of bring in another concept that we had learned, which is actually using a reporter fusion to a PIL-U ATPase that is involved in this pilus formation and energizing to have this happen. So they've used it to GFP, and so you can see the cells that are expressing PIL-U during this twitching process. So they'll be green. So you can see that not all cells are expressing high levels of it, but some are. Okay. Now, one last video. Actually, two. I'm going to do two. Okay. So this is from a friend of mine, Professor John Kirby, who's at the Medical College of Wisconsin, and he works on a bacteria that actually preys on other bacteria, which I think is super cool, right? So it's called Myxococcus xanthus. It's a soil-dwelling microbe, and it can like annihilate colonies of other bacteria by eating them or being predatory on them. And so a lot of it is mediated by type 4 motility, so it will crawl towards its prey. And then as soon as it hits the prey, it starts to ripple in the colony, which is also mediated by coordinated communication process, and then it will just dissolve it, okay? So he has time-lapse videos of this Myxococcus xanthus colony preying on, oops, not this one, sorry, an unsuspecting E. coli colony. Okay, so this is Myxo on the left, and this is E. coli on the right. And you can see that it's twitching and moving, and once it hits the E. coli colony, it starts to do that rippling, okay? So this is something that is being heavily studied in labs that work on microbial development, especially in Myxococcus xanthus, like what signals those waves? And it's the predation waves, and then it just like eats the E. coli, and then it spores. And there's actually some research on this. So it does this to many bacteria, not just E. coli. And so there's some research trying to look at it in terms of like, could you put this in a wound dressing or something where it could be used in a clinical setting? And I've always wanted to look at it in terms of plant disease control, but haven't yet got to it. Okay. So this is just the video of the waves close up, and I think these are nice. So this would be like if you were zoomed in on that Myxo colony eating the E. coli. And it only does this wave formation when it is in contact with its prey. And so it's some sort of communication process. Yes?
Do you have flagellar motility when you have like okay, I'm trying to remember my question. Okay, if you have like flagellum that's eating like toxin and
Yeah.
like towards a specific like food group or something that speeds it up.
So like chemotaxis?
Yeah, chemotaxis. If
Mhm.
you have something like that, is it similar to the switching motility of type 4 pili? Like, will it be directed toward a specific location?
Yes, so there is she's asking is chemotaxis involved in twitching motility like this, such that we described in swimming motility for flagella? The answer is yes. There are a lot of chemotactic proteins and processes involved in twitching motility as well. Okay, so that was twitching motility. And I know you're just all enthralled now. You can go look at all of these videos online. So, to remind you of how this sort of links to biofilm formation. So remember, bacteria attach to a surface and then they need to crawl towards each other and form those microcolonies that then eventually form biofilms. They do that crawling through twitching motility. So that's the surface-based motility. Okay, so now switching gears and talking about non-pilus adhesins, of which there are many, 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 many. And your textbook has pulled out these two as examples. One is called M protein, which is produced by Streptococcus pyogenes. And you can see that you have these fibrils in the left panel here around the surface of the cell, and they mediate attachment to host tissues for this bacterium. Bordetella pertussis, you'll see on the right, that's the causal agent of whooping cough, and so it colonizes the trachea here. And you can see that these bacteria are intercalated into or near the cilia of the trachea, and this attachment is mediated by at least one adhesin called protactin. Okay, so biofilms and infection, just to kind of circle back to that. So biofilms, like any surface, is vulnerable to attachment of bacteria and eventual formation of a biofilm, and they're very, very tightly correlated with pathogenicity. Okay, so sometimes if you obliterate the ability of a particular organism to form a biofilm, it is not a very good pathogen anymore. Okay? So they can play an important role in chronic infections. So if you think about someone who has osteomyelitis or a wound infection or something that's colonizing an indwelling medical device, so they really like to form, bacteria really like to form biofilms on metal. So a lot of things that are in your body, so if you have a knee replacement or something like that, there can be metal parts to that, and that can be very problematic if that becomes colonized by an antibiotic-resistant biofilm. And so trying to treat that with antibiotics can be tricky, and sometimes patients have to end up removing the piece of metal that is part of their knee replacement. So this actually happened to my father. So he broke his kneecap, and they didn't replace it, but they had to like piece it back together with wires. And he got osteomyelitis in that knee, and so they eventually just like, okay, we're going to take out this wire because it's causing problems. And so sometimes in a clinical setting, you have to sort of weigh the risks and remove the thing that's causing infection. And so he had methicillin-resistant Staphylococcus aureus infection, which is a MRSA, and they had to really aggressively treat that with antibiotics, and then do something like remove the metal piece in his knee to get rid of that, okay? So chronic infections can be really problematic because these bacteria are living in this community of cells. Some of them might be dead, but the internal ones are alive. And so if you are trying to treat that with antibiotics and you can't penetrate that thick biofilm, there's always going to be some living portion of that population, which can be very hard to treat. I'm sorry, this is bothering me. I'm going to ask you. So this can be important for otitis media, which is the inner ear infection. Also, we talked about dental plaque being important as a biofilm component of your mouth. And then other things like chronic wound infections or infections of cystic fibrosis patients where they have these chronic Pseudomonas aeruginosa infections in them. So like I mentioned, these biofilms are often resistant to antibiotics. They're sometimes very hard to clear from hosts. And so there's a lot of research being done on trying to understand how biofilms are built, how can we disassemble them, and how can we develop antibiotics that would either disrupt that process or penetrate those thick biofilms. So these are just some pretty pictures. This is a biofilm on an infected tonsil. So a lot of times in nature, these bacteria or these biofilms are multi-species, okay? So that can also be a problem. You're not trying to target just one species of a microbe, you have many inside of this. And then right over here on the right is a panel of an infected adenoid from a pediatric patient. And so it was covered in different bacteria of different species, and you can see that they're in this robust biofilm. Okay, so we will leave off there. We're going to continue next lecture on microbial pathogenesis part 2. So we're going to talk about toxins and how they're secreted.